Why does weight so often return after stopping semaglutide, tirzepatide, or after bariatric surgery? The answer is definitely not a lack of discipline. After major weight loss, your body may retain a powerful metabolic memory, one that directly affects your appetite, energy expenditure, fat cells, the gut microbiome, mitochondria, and even the way your genes are regulated. Welcome to this explainer. We're going to dive right in and unpack the deep biology behind the yo-yo effect.
Because here's the crucial point. Weight loss is only the first phase. The real biological challenge is metabolic stability. When the pharmacological effect of an incretin treatment declines or that initial shock of bariatric surgery passes, your body actively works to return to its previous set point. Today we're exploring exactly why that happens and, more importantly, how targeted, multifaceted nutritional strategies might actually support your transition back to long-term stability.
So here's the roadmap for our explainer. 1. The biology of weight regain. 2. Gut signaling and incretin defense. 3. Cellular resilience and polyamines. And 4. Muscle and metabolic stability. Let's get right into it.
All right, part 1. The biology of weight regain. Let's understand the yo-yo effect. When you lose weight, your body responds biologically. Your appetite rises, that constant food noise returns, leptin signaling drops, and your resting energy expenditure actually stays reduced through adaptive thermogenesis. But crucially, we've got to distinguish between how fat is stored. On one hand, we have metabolically healthy obesity, or MHO, which is mostly subcutaneous fat with lower inflammation. Contrast that with MUO, metabolically unhealthy obesity. MUO involves visceral fat packed tightly around your organs, enlarged fat cells, insulin resistance, and chronic inflammation. If a person had MUO before losing weight, that less favorable, inflamed metabolic environment makes the rapid return of visceral fat so much more dangerous when those excess calories come back.
Now, weight loss usually shrinks the size of your fat cells, right? But it doesn't eliminate most of them. Recent research, like the incredible 2024 findings in Nature by Hinte and colleagues, shows that adipose tissue actually retains a persistent epigenetic and transcriptional memory of obesity. The DNA and histones inside these fat cells literally remember their previous state. Basically, your fat cells hold a biological grudge. Now, this doesn't mean you're permanently programmed to regain weight. It is absolutely not an irreversible destiny. But it perfectly explains why previously obese tissue responds so rapidly to a caloric surplus.
And it's not just genes. There's an immune memory at play here, too. See, adipose tissue is a highly active endocrine and immune organ. Research from Cottam in 2022 showed that even after weight loss, a residual immune sensitization sticks around. Pro-inflammatory macrophages and T cells are basically still hanging around those shrunken fat cells. So when energy returns, it can quickly trigger renewed inflammation, mitochondrial stress, and insulin resistance.
Moving on to part 2. Gut signaling and the incretin defense. It all starts in the gut. When food reaches your intestine, enteroendocrine L cells release natural GLP-1. This is fantastic. It supports satiety and improves post-meal glucose control. But look closely at step 3 here. GLP-1 secretion is really only half the equation. In our bodies, natural GLP-1 has a very short active life because an enzyme called DPP-4 rapidly cuts and inactivates it. So to support the incretin access nutritionally, we would theoretically want to stimulate natural GLP-1 and perhaps modulate that DPP-4 enzyme that degrades it.
And this is where advanced nutrition comes into the picture. When proteins like milk whey are broken down through kefir fermentation, or when soy proteins are fermented, they generate these smaller bioactive peptides. Experimental enzyme studies suggest that some of these peptides are capable of interacting with the DPP-4 enzyme. Now, to be crystal clear, this is a proposed nutritional mechanism. It is not a pharmaceutical equivalence to a DPP-4 inhibitor drug. But potentially, these food-derived peptides may modulate DPP-4, allowing your natural incretin signals to stick around just a bit longer.
Alongside proteins, we've got fermentable fibers. When fibers from sources like sorghum, cruciferous vegetables, and mushrooms reach your colon, beneficial bacteria ferment them into short-chain fatty acids, or SCFAs, like acetate, propionate, and butyrate. These SCFAs interact directly with intestinal receptors, specifically FFAR2 and FFAR3, to naturally stimulate your L cells to release more GLP-1 and PY. It is a beautiful, natural partnership between your diet and your microbiome, supporting satiety and a really healthy gut barrier.
Okay, part 3. Cellular resilience and polyamines.
I want to introduce you to a truly fascinating ingredient called the Amrita complex. This is SCOBY fermented bee pollen. Bee pollen has a highly resistant outer shell,
almost like microscopic armor, but the fermentation process disrupts this shell, releasing an incredible profile of amino acids, polyphenols, and notably polyamines. Analytical lab evaluations from Medica SRL labs have identified highly bioavailable polyamines in amrita, specifically spermidine and conjugated spermine. And these molecules, they add an entirely new layer of cellular defense.
So, why do we care about spermidine? Well, it comes down to autophagy and mitophagy. These are basically your cell's internal recycling and maintenance systems. They clear out damaged proteins and dysfunctional mitochondria. Spermidine is heavily involved in supporting these systems. It doesn't directly stop weight regain on its own, but it supports the cellular maintenance that helps your metabolic tissues, like your muscle, liver, and fat cells, adapt and remain functional under nutritional and oxidative stress.
And that brings us to a major cellular threat called ferroptosis. Ferroptosis is a regulated form of cell death driven by iron-dependent lipid peroxidation. Let's be totally clear here. This isn't the primary cause of the yo-yo effect, but the cellular stress and damage caused by ferroptosis heavily contribute to metabolic dysfunction in your liver, pancreas, and adipose tissue. If your cells are dying from oxidative stress, your metabolism simply cannot remain stable.
This is where the spermine we just mentioned comes into play. Groundbreaking experimental research, like the 2026 Lee et al. Study in Nature, suggests that spermine can function as an endogenous iron-binding molecule. By interacting with labile, or free iron, within the cell, spermine may reduce what are called Fenton reactions. Fewer Fenton reactions ultimately mean less lipid peroxidation, offering greater protection for the cell membrane. The conjugated spermine in the fermented formula acts as a molecular reservoir for this exact protective pathway.
However, we've got to look at the full picture here. Polyamines actually have a dual, context-dependent nature. On one hand, you have the protective pathway supporting autophagy and iron homeostasis. But on the other hand, excessive catabolism of polyamines can actually generate reactive aldehydes and oxidative stress. So polyamines are not some magic, universal block against ferroptosis. Their ultimate effect depends heavily on dose, tissue health, iron availability, and overall cellular balance.
And finally, part 4, muscle and metabolic stability.
Listen, weight maintenance requires so much more than just staring at a lower number on the scale. It's about preserving skeletal muscle. Muscle is your metabolic engine. It supports massive glucose uptake, drives insulin sensitivity, handles fatty acid oxidation, and dictates your resting energy expenditure. You absolutely must combine nutritional support with adequate dietary protein and resistance training to preserve this crucial metabolic machinery.
When we put this all together, we see 4 integrated mechanisms provided by this fermented nutritional formula. Number 1, supporting physiological incretin secretion. 2, the possible modulation of the DPP-4 enzyme by bioactive peptides. 3, profound support for the microbiome and intestinal barrier. And 4, cellular and mitochondrial resilience driven by polyamines like spermidine and spermine. These don't operate in isolation. They interact rapidly and synergistically in a beautiful systemic symphony to support your metabolic stability.
Now, I need to be incredibly clear about this. This multimodal nutritional approach is not a replacement for medical treatments like semaglutide, tirzepatide, or bariatric surgery. You should never change your medication without consulting your prescribing physician. What this formula represents is a potential nutritional bridge, a scientifically rational support system designed specifically for that delicate transition from active weight loss to long-term metabolic stability.
As we synthesize all of this, it becomes so apparent that the yo-yo effect is not driven by one single hormone, one missing enzyme, or a simple behavioral failure. It is a massive biological cascade. It emerges from a complex interaction between rising appetite, lowered energy expenditure, the epigenetic memory of your fat cells, renewed inflammation, the risk of muscle loss, and severe mitochondrial stress.
And that is exactly why a targeted nutritional approach needs to be equally complex. By combining kefir fermented whey, fermented soy, sorghum, cruciferous vegetables, medicinal mushrooms, wheat germ, and the amrita fermented pollen, you're addressing several of these biological pathways simultaneously. From incretin signaling to iron homeostasis, this precise blend of varied fermented ingredients offers a powerful, scientifically grounded rationale for fighting back against the yo-yo effect.
Weight loss is a monumental achievement, but keeping it off requires changing the biological environment of your body. From the GLP-1 and DPP-4 access to the microbiome, from preserving muscle mass to driving autophagy and protecting against cellular lipid peroxidation, we are truly looking at the future of metabolic support. While clinical studies on the final finished product will be the ultimate test, the science of these mechanisms gives us an incredible roadmap. We can no longer view weight maintenance as a mere test of willpower, it's a test of cellular strategy. So the question is, are you equipping your metabolic machinery for the long haul? Thanks for joining me on this explainer, and keep learning.